Welcome, everyone, to the N-of-One Collaborative Seminar Series. My name is Nicole Nolan. I'm N1C's Programs Manager. We're very happy to have you attend on this post-US holiday Monday. To kick us off, I'd like to first and foremost thank N1C's partners, the Chan Zuckerberg Initiative, Hanjin, and Kim Jeans. We are so grateful for their support. If you would like to discuss how your organization can help support N1C's mission, please reach out to me. I'll put my email in the chat shortly. I also want to mention that N1C has recently launched a new online platform called Connection that supports collaboration across the individualized medicine community virtually. So we will be continuing discussion from today's seminar on connections. If you have any questions that pop up after today's meeting ends, please feel free to post them there and we'll make sure that you get an answer. With that, I will turn the rest of the meeting over to Dr. Marlene Laufer.
Thank you so much, Nicole. Hi, everyone. I'm Marleen Laufer. I'm a postdoc at the Dutch Center for RNA Therapeutics, and I'll be moderating today's session. And there's a few housekeeping rules from me today. So we'd love this to be an interactive session. We'd love to see your faces, so please feel free to turn on your cameras so we can see you. But please be reminded it is recorded, so if you do not want to be on camera, of course, feel free to leave the camera off. As Nicole said, the session is recorded and will be available on Connections afterwards, and Christiana, our speaker, has kindly agreed to be able to answer your questions there as well. If you do have questions today, immediately after the presentation, please put them in the chat. I will moderate them and then bring these questions forward to our speaker. And at the end of the session, you'll receive an automated survey via Zoom to be able to evaluate the seminar. So we'd love to hear your feedback and any recommendations for other seminars we might be hosting in the future. And with that, I'm done with the housekeeping, and I'd be very happy today to introduce to you Christiana Wang. She's a PhD candidate at Baylor College of Medicine in Texas. She's working in Pengfei Liu's lab, and Christiana has been doing a wonderful job for the N-of-One community in the past 2 years and has been presenting her data also at our conferences. And today she's been going to talk about the SPTAN work and an ASO development for these patients. So Christiana, please take it from here, and we are all very excited to hear what you have to tell us.
All right, thank you so much for the introduction. I'm gonna quickly share my screen and let's bring that to full screen. All right, is everything good? Slide-wise, everybody can see it? Good, thank you. I'm gonna bring up my laser pointer. All right, okay. Thank you for the invitation again from N1C, and I'm really happy to share some of our research on SPTAN1-associated disease mechanisms and the potential therapy in patient-derived cell lines. So I'm going to quickly do an outline for today's talk. So I'm going to start focusing on SPTAN1 mutations and DE5 and the potential therapy that we've done quite a bit of research on. And moving on from that, we're going to look at some bigger pictures coming from a research and a diagnostic lab that I'm in, and what we've learned from this particular project to how we can broaden our perspective to enhance the workflows. So to first start off by focusing on SPTAN1 itself, I'm going to quickly introduce this particular protein to you. So SPTAN1 encodes for alpha-2 spectrin. It is a non-erythrocytic scaffolding protein subunit that interacts with other forms of beta spectrins by forming a heterodimer, and then that heterodimer forms a tetramer. And this particular protein sits right underneath the cell membrane, and it interacts with ankyrin protein, which allows the binding for ion channels in various tissues. And this particular gene is expressed ubiquitously amongst our body. So if we want to focus more on the relevant clinical phenotypes, we wanted to zoom in on alpha-2 spectrin's participation in neurons and in the central nervous system. So this illustration here sort of depicts the structure of a neuron. And if we think about where alpha-2 spectrin is, they're indicated here by these blue little bars. So they form tetramers with other forms of beta spectrin. And I do want to focus more on the axon initial segment here. So this is where the axon starts to form. And in particular, at the axon initial segment, alpha-2 spectrin interacts with beta-4 spectrin. So this beta-4 spectrin marks the axon initial segment and is uniquely expressed only in neurons and only at the axon initial segment and at the nodal gaps. This spectrin tetramer allows for the binding of ankyrin G protein, so it is a secondary interaction to the spectrin tetramer, and then the tertiary interaction would be your sodium channels, which allows for the signal propagation in the neurons. So when we go back and look at SPTAN1 and its associated Mendelian disorders in general, we do notice that SPTAN1 is associated with various severity of clinical phenotype presentations, and we've done a literature search for the case reports that are out there for SPTAN1 associated disorders. And we've categorized them into 4 different categories, ranging from the most severe to the least severe here encoded by the different colors, with red being the most severe with risk of early mortality, and green being the least severe with more milder movement disabilities. And we've sort of organized them here along the gene, and we noticed that there are hotspots for these most severe clinical phenotypes located within the dimerization domain. And we also see that when we put the mutations that are either frameshift or nonsense here on top of the gene, we notice that they're mostly in the milder clinical forms, and this gives us suggestions about the potential disease mechanism of the variant that we're focusing on, which is this duplication variant here located within the dimerization domain, and we're suspecting it to be either gain of function or dominant negative because it is much more severe compared to those that have a loss of function consequence. So because we have this stipulation, we go on further to look at previously published data on cellular phenotypes from patient fibroblast lines. So this is a paper published in 2017. They basically looked at the control cell lines. This is patient fibroblasts, looking at interaction between alpha-2 spectrin and beta-2 spectrin, so one of the beta spectrins. And you see that here from patient samples with the particular duplication variant, there is significant aggregation between the alpha-2 spectrin and the beta-2 spectrin here. And since we know that the spectrums are ubiquitously expressed, so it is very much expected that fibroblast cell lines would have this representation. And then another paper back in 2010, where the mutation is first discovered, took a step to utilize expression plasmids that either express human wild-type SPTAN1 or a SPTAN1 with the duplication variant. And they looked at the interaction between the expressed human SPTAN1 with the mouse ankyrin G. So these are all done in mouth neurons. So in a wild-type situation, there is basically not much difference in terms of the axon initial segment here, marked by this dashed line, or here by the red signal indicated by the ankyrin G, but in a situation where the duplication variant is expressed, you will see that duplication variant deforms the neural morphology, and there is a shortened ankyrin G signal or axon initial segment length, and that is an indication of axon initial segment destabilization. So there's 2 lines of evidence for the cellular phenotypes previously reported in the literature. So we took these expression plasmids and decided to look at the primary interactor of alpha-2 spectrin. So ankyrin G, again, if you're reminded, these are the secondary interactors that play a part at the axon initial segment. We decided to look at the beta-4 spectrin, which is the direct interactor with alpha-2 spectrin. So we basically took the expression plasmids the previous paper has used, and we did basically overexpressed them in rat hippocampal neurons and looked for positive signals of that expression plasmids. And in a wild-type situation, you see that the beta-4 signal is very clearly indicated here, marked by the purple line. And it is very straightforward. It is a line of axon initial segment. But if you introduce the duplication variant, you will see significant aggregation between beta-4 spectrum and alpha-2 spectrum, and they're starting to form aggregates in the somatodendritic region instead of elongating along the axons, and that indicates the defects we are expecting caused by the duplication variant. We brought this even further. So all the data that I'm showing you on the neuronal level are either in rat neurons or in mouse neurons through an expression plasmids. We wanted to see that happen real time in patient-derived cells. So we did obtain neurons differentiated from patient cell lines, and we decided to look at the interaction between beta-4 spectrum and alpha-2 spectrum. So on top here, we're looking at a healthy individual. A proper AIS is marked by this white arrowhead here, and you can see here in a healthy individual, we see the AIS in a straight line, and in affected individual with which we used here, subject B in our cohort, we see an aggregate here marked by the red arrowheads, and the blue arrowheads are marking the disrupted axon initial segment. And if we zoom in further to these specific loci, we will see the colocalization differences from between a healthy individual versus an affected individual. So on top, you're looking at the colocalization analysis for this particular segment here along an axon initial segment. The purple color indicates for beta-4 signal expression, and then the green signal here indicates for MAP2 signal expression, and the MAP2 is a dendritic marker, where purple is a beta-4 marker, which is the axon marker, axon initial segment marker. So we define axon initial segment disruption through the invasion of MAP2 signals, or a dendritic marker signal into an axon region. So where a healthy individual has no overlapping signal between a dendritic marker and an axonal marker, in a disrupted axon initial segment, you see this great overlap between the dendritic marker and the axonal marker. So this is a quantifiable way that we can define axon initial segment disruptions. So beyond beta-4 spectrum, we also wanted to look at other beta spectrums that alpha-2 spectrum interacts with. So in a healthy individual, the signal from SPTAN1 actually encodes for beta-2 spectrum. I know the numbering system is kind of weird, but beta-2 spectrum interacts with alpha-2 spectrums at the somatodendritic region. So you can see this diffused signal in a healthy individual. In an affected subjects, you will see these red arrowheads are pointing to the aggregates that are forming in the somatodendritic region, and this is happening within every single neuron that you will see. So this gives us an understanding that it may not be gain of function, it particularly could be dominant negative or toxic against all spectrums. So additionally, because everybody in the field previously have sort of looked at anchorin G signal, which is the secondary interactor to the spectrum tetramers, we also decided to just take a look at that. So in a healthy individual, your anchorin G signals are basically sitting at the proper axon initial segment, overlapping with your beta-4 spectrum. And in an affected individual, your beta-4 spectrum is obviously aggregating, and then your anchorin G signal is either absent or very weak compared to the healthy individual. So this basically kind of gives us a good understanding of what the expected neuronal phenotypes are coming from a patient-derived neuron cell lines. So going back to that quantification that I sort of mentioned with AIS disruptions, we can define AIS disruption with these two markers. So a healthy individual with no AIS disruption would have a positive beta-4 signal and a negative MAP2 signal. And then a disrupted AIS would have a positive beta-4 signal and a positive MAP2 signal. And we did our initial analysis, a statistical analysis based on the data that we currently get, and we see a very significant differences between the wild type, which is the healthy individual, versus the duplication variant carrying affected individual. So with that all figured out, we are expecting a very clear disease mechanism based on the molecular phenotypes, as well as again mentioned going back to the clinical phenotype, where the disease severity is much more severe compared to the other categories based on their loss of function variants being less severe compared to the one that we're interested in. So there are two lines of evidence that basically gave us an understanding that we are looking at and dealing with a duplication variant, and this does affect the way that we approach this particular disorder in terms of what ASO we should use and what ASO strategy is appropriate and to what degree is safe for the patients. So the two lines of evidence, as I mentioned before, the clinical phenotype is more severe than the loss of function variants. And then the cellular phenotypes that we are looking at demonstrates aggregation in the neuronal lineages. So with that, we can say, obviously in a normal situation, your two alleles of SPTAN1 is producing your wild-type protein. The duplication variant here on a single allele will create a dominant negative phenotype, which leads to a severe clinical phenotype. And if we wanted to restore for function, we are looking at using an RNase H-based ASO, which is a gapmer ASO that can target the duplication variant or the duplication variant carrying allele to remove that particular mRNA, utilizing the endogenous RNase H mechanism in our body. And then hopefully we can remove most of these dominant negative proteins, toxic proteins, so that we can restore to, in the best situation, 50% wild-type expression. But that is not considered a cure for our patients because when we go back and look at the clinical phenotypes, even the patients with loss of function variants are suspected to milder forms of disease. So, but because of our patients are with higher risk of early mortality and significantly more severe seizure phenotypes, we believe that approaching the treatment options with gapmer ASO would significantly alleviate their disease burden and better their quality of life. So that's kind of all the logistics behind how we wanted to approach the SPTAN1 dominant negative disorders. So going forward, we are actively recruiting patients worldwide. So if any of the clinicians on the call have encountered patients with SPTAN1 duplication variants, we are happy to accept them into our program. Currently, we've recruited 7 patients from 6 families in 3 different countries. We began our recruitment in early 2022. That's actually happened before I joined the lab. 5 out of 7 patients with the duplication variant are still alive as of today, which reflects the disease severity that we're dealing with, and we're working against time for our patients. And our oldest patient alive today is around 6 years of age, and that's kind of the average expected lifespan for patients affected by this duplication variant. So going into more details of the actual variant itself, here on the graph, I've basically pointed out what the variant looks like. So it is called a duplication variant, but in reality, it is a small indel or a small insertion of 9 base pairs, so here indicated in red, compared to the wild-type allele. But originally in the wild-type allele, these 9 base pair is already duplicated, so you're looking at an existing in tandem repeat of 2 times 9, which is 18 base pairs that already exist in the wild-type allele. And in the variant allele, you would have 3 times 9 of that, which results in a 3 amino acid addition. This actually proposed some problems for us. So first of all, if you design an ASO within this region, you are basically spanning across 3 in tandem repeats, and that results in hairpin structures for ASO itself. So using the oligoanalyzer tool from IDT, you will see that the ASO itself will form hairpins. So that significantly reduces the efficacy of your ASO. And secondly, using the dimer predictions, you will see that the ASO actually forms pretty stable oligodimers. So instead of binding to the targeted mRNA, the ASOs could be just simply interacting with itself when it's in solution. So both of them produce significant problems. And when we tested that in our patient cell lines using transfection reactions, we noted that there weren't any significant or effective knockdown across 2 patient cell lines. And you can see that is very likely contributed by the reasons that I explained above. So an alternative strategy that we've considered is utilizing linked SNPs, so linked single nucleotide polymorphisms that are along the same allele as the disease allele. So by knocking down a linked single-linked SNP, will be able to bring down the expression of that entire allele. So any heterozygous SNPs along this entire gene, as long as we can phase them to be on the same allele as the disease variant, we will be able to use them as a candidate target. So we've completed long-range sequencing for 2 of our patients up to date. Actively working on our other patients. So based on the information that we received, we identified some lead candidates from these patient cell lines based on the local sequence pattern. So if you have quadruplet Gs, like something that in the community that we know of are not good ASO candidate regions, we've eliminated them. So the legends here are basically pointing to exonic SNPs, indels, or intronic SNPs, and we do note that with SPTAN1, there is majorly intronic SNPs available for targeting. So because there is a lot of SNPs, and as a research lab, we don't have the capability to screen through all of them. I've designed a small prioritization tool to help us pick candidate ASOs that will have a better accessibility based on secondary structures. So I decided to obtain the FASTA file for 100 base pairs of mRNA information centered around the SNP target. And there are two informations that I require. So first of all, I tile the ASOs by positioning the SNP starting from position 6, which is the gap region for a 5-10-5 MOE ASO. And then I'm going to look at GC content, and I've defined acceptable GC content range between 35% to 65%, which is commonly accepted in our field. And secondly, I wanted to look at secondary structures. So I decided to use the Vienna RNA fold software to predict secondary structure scores. So an unbound nucleotide, which would be located within a loop. A bound nucleotide would have an interactin partner on the mRNA, so it will be in a stem. So we basically define based on previous literature's data, like scattered everywhere, that sometimes in a gap region, the unbound nucleotide for accessibility of RNase H would be beneficial. In the modified region on each flap ends, a unbound nucleotide for accessibility is critical for the recognition of your ASO to the mRNA sequence. So I decided to score those regions higher than in the unmodified gap region. So you could have a bubble for your ASO where both ends of the modified regions are bound, but the gap region here is unbound. So um, basically across 20 nucleotides, each nucleotide would have a score defining whether it's a bound nucleotide or unbound nucleotide. Unbound meaning it's more accessible, bound meaning it's less accessible. So making that score a positive score for unbound and a negative score for bound, I'm able to get a accessibility score for each of the ASOs that I've generated that passes the GC content filter. And with that, we can basically do a ranking system for ordering our ASOs. So we decided to get minimum of 2 ASOs per SNP so we can do our initial screening across the ASO candidates that the SMP candidates that we've identified. But this seemed really promising for me as a scoring system ahead of time. But when we actually go back and see our results and combine our data and compare it against significant knockdown versus non-significant knockdown, we don't see any correlation between the secondary structure score or the GC content. So regardless of the GC content being high or low, but both of the knockdown efficacy, they're roughly around 50% of GC content. And with secondary structure scores, although the significantly knockdown ASOs have a slightly higher secondary structure accessibility score, they're not much different from the ones that are not creating a significant knockdown. So again, this is just open to the community, I guess, for discussion is what are the features that allows for ASO efficacy, if there's any software predictions that allow us to better the computational predictions prior to manual screening. So that's my attempt, and it basically reflected that secondary structure and GC contents on my end don't really correlate to ASO efficacy. So going on to the actual screening itself, we performed our initial screen using patient-derived induced pluripotent stem cells. And with iPSCs, the ASO has to be transfected into these cells. So here's kind of an overview. There's a lot of information on this slide, but you don't have to look in detail. So on the left here are the mutant allele expression for one of the subjects that we screened. The wild-type allele expression, everything is normalized to a percentage compared to the scramble control. So basically at 1, so at 100%, you're looking at a scramble control expression. So the negative C2 here, the purple line all the way to the right. Anything that is lower than 100%, we're looking at knockdown. So the arrows here are pointing at significant knockdown in in blue, and then allele-specific significant knockdown. So mutant allele being significantly knocked down, where the wild-type allele is not significantly knocked down, are pointed here by the green arrows. So we've identified around 11 ASOs for one of the twin subjects that we've screened. So you can see that at 50% expression here, marked with the dashed line, most of the ASOs that we've gotten are non-allele specific based on transfection results out of the 52 ASOs that we screened. And with the other subjects that we had long-range WGS available, we've screened through similar methods using ASO's transfection in iPS cells. And again, we've identified a few candidate targets with significant knockdown in either both alleles or in the targeted mutant allele. So out here, we had 8 ASO candidates that had either allele specific or non-allele specific knockdown compared to our scramble control out of the 50 ASOs that we've screened. So we decided that beyond the large scale screening that we've done with over 100 ASOs that we screened across two patient cell lines. We wanted to test the top candidates through genetic delivery, which means passive uptake in the patient-derived neurons. And the reason for that is obviously we're looking at central nervous system phenotypes with the seizures that are significantly affecting our patients. So that testing the delivery in neurons would be lineage specific to the disease. So in patient-derived neurons, we tested for dose response curves for our lead ASOs. So we've only have data for 2 ASOs per patient right now, but I'm gonna quickly show you what that looks like. So the blue line here, which indicates the C allele, the purple line here, or the pink line here indicates the T allele. And for subjects A1 and A2, the blue allele is the mutant allele, and the T allele is the wild type allele. So you're looking at, we're looking to knock down the C allele. So the first candidate ASO, while it has shown significant knockdown in transfection reactions. We're seeing knockdown, but not allele-specific knockdown, all the way up to the highest dose here, which is, I believe, 10 micromoles in genetic delivery. And for the second candidate ASO that we've tested, all the way up to 15 micromoles genetic delivery, we are seeing some allele specificity, but we're not getting the expected efficacy for this particular ASO. So that just kind of reflects we need to test more candidates for this particular subject. So going back to our list of 11 top candidate ASOs with knockdown in our initial IPS screen, we'll be able to do more testing in genetic delivery in neurons. But for our other subjects, so subject D, we did identify a lead candidate. So ASO number 69, obviously, we can see that it is not the desired level of knockdown. I do want to point out again that in this particular subject, the C allele is the wild type allele, the T allele is the mutant allele that we're targeting. So they actually do share the same SNP for measurement, but the disease allele is on a different allele. So for subject D, we had a lead candidate ASO here, number 67, where we are getting a dose-dependent knockdown for the mutant allele while sparing the wild type allele. So this one will be further assessed for improvements in efficacy. So we're hoping to bring down the mutant allele expression as close to 0 as possible because it is a dominant negative disorder. So any amount of toxic protein left in production will likely interfere with the wild type's protein's function. So we do want to reflect on the different delivery methods that we've utilized in this screening process. So they each have their advantages and disadvantages in this screening that we've done. So we started off with the bulk large-scale screening with patient-derived iPSCs transfected with the candidate ASOs. The advantage to that is obviously it's a shorter time frame. IPS do grow a lot faster, and we do need less ASOs for transfection reactions. So we're looking at so the experiments that we've utilized 200 nanomolars of ASOs each. The disadvantage to that is you're adding a second step to the delivery systems. You're utilizing transfection reagents, and that efficiency may actually vary between experiments or between different wells of application. So that causes some instability in the data that we generate. But if you consider the utilization of IPS differentiated neurons, you're basically getting the reversed advantages and disadvantages. So the culturing time will take much longer for IPS differentiated neurons, which is 3 weeks minimum for our protocol. And we do need more ASOs for this type of passive delivery. You're looking at a scale starting at 100 nanomolars, going all the way up to 15 nanomolars, which is the range that we've utilized in our research. But the advantage to that is because you're eliminating a step of transfection, you do get more consistent results between your replicates, either different batches or across technical replicates within the same batch, because you're eliminating a factor for transfection in terms of delivery. So with that, that basically kind of concludes what we're at for our SP101 projects. And we're actively working on optimizing our lead candidate ASOs and performing some of the functional analysis in terms of the axon initial segments that I've mentioned previously in terms of their disease mechanism, seeing if we could restore a proper non-disrupted axon initial segments. So I wanted to step back a little bit and reflect on what we're learning from this project, in particular within our laboratory, which is a research and a diagnostic laboratory working side by side with each other. So when we looked at our timeline for the SP101 project, we do note that there is a significant amount of time that elapsed between the first sample we received for our patients all the way up until today, where we're at the point where we have some lead candidate ASOs that we're looking to optimize. So there is a significant amount of time that we spend on troubleshooting or dealing with the challenges that the mutation itself has proposed in terms of that tandem repeat region and trying to figure out proper disease mechanism and evidence for any functional baselines that we can utilize to assess for therapeutic benefits. But if we do focus on some of the later subjects, once we had that part figured out, it took us around 18 months between the time of receiving the sample to the point of a lead candidate identification. So, and that time is going to get shorter and shorter as we are more comfortable with a particular gene, knowing the disease mechanism and the approach that we're taking for ASO developments. And if you consider the timeline that Tim Yu's lab, which is the pioneer for N-of-1 clinical trial development for using ASOs, they do have a clear disease mechanism. So by identifying the deep intronic variant in the gene, they basically understood that it was a cryptic splice site leading to the disorder. And it took them around a year for preclinical development to happen to the point where they have to the point where they are ready for toxicology study, leading to IND enabled IND filing and first patient dosing. So sometimes splice correcting ASOs on that end are easier or quicker to establish. But with our experience in our lab, we've identified that additional personalized ASOs for other genes that when patient families approach us, will take much lesser time for us to develop in the future, depending on the available information that has been previously published. So cell line generation is sort of a restricted time frame. It takes around 3 months for us to get to the lineage specific cell types. For functional analysis, this can vary very much because you either have published information that are available that you just need to replicate in your own laboratory, or you need to figure out disease mechanism like what we've done for SP101. ASO design and manufacturing can take anywhere between 1 to 2 months, depending on where you order ASOs from, and then the large scale ASO screening will take between 2 to 3 months, depending on either you're screening with transfections or you're screening with passive delivery. The lead candidate ASO optimization can take multiple rounds, so this will be a longer time frame that's required compared to our large-scale ASO screening. And for us, the functional rescue is a very important step. So based on the functional baseline that you established previously, either utilizing published data or utilizing disease mechanism study established in your own lab, that functional rescue is going to indicate for us the actual therapeutic benefit and the required amount of change that's needed for any functional differences, at least on the cellular phenotype levels. So as a part of a diagnostic lab, we wanted to say that reflecting on our experiences dealing with clinicians, patient families who have come to us with a genetic or clinical diagnosis, and asking for help for N of 1 treatment options, we realized that this could be an isolated and fragmented process in the pipeline. So currently, the protocol looks like you have clinicians, patient families, and diagnostic labs that come up with the genetic and clinical diagnoses. And then the patient families are basically left with their diagnoses and they're required to look for their own options. So there's a large disconnect between the options for treatment, and many families are unaware of or are ineligible of N of 1 trials that are out there that are hosted by nonprofit or philanthropy development organizations. So what we as a diagnostic and research lab wanted to offer is theoretically something a little bit better. So instead of leaving patients with a simple genetic diagnosis, we wanted to think about the options of, as a diagnostic lab, do some preliminary screening, analyze treatment options, and bring that report to clinicians so that when clinicians are reporting the diagnostic results to the patient and their families, they can also educate them with N of 1 treatment options for these families so that when they come back with their results, they know their options and the resources to look for in the drug development processes. And how that looks in detail is traditionally, as diagnostic labs, we provide DNA sequencing results or RNA sequencing results. But the diagnostic labs are equipped with personnel that could utilize the published N1C guidelines for assessing therapeutic eligibility. So that could be information that's included in a report that's provided. And additionally, because sometimes diagnostic RNA sequencing will require a lineage-specific differentiation for patient cell lines. And with that information available, we can do transcriptomic analysis or even have the patient cell lines ready for researchers who will be able to, or researchers or companies who will be able to take on the actual drug development process. So what that looks like on the clinician's end is that they will have a new diagnostic lab report that has additional information. So beyond genetic diagnosis, they may have transcriptomic signatures that are available to provide to subsequent researchers or companies. They will have information for the patients in terms of their therapeutic eligibility and their recommendations, and the relevant cell types for diagnosis and drug screening if that becomes available and a research group or a company is willing to take on the N of 1 development. So it seems like a very promising or a active role of development for diagnostic labs in academic environments that will help advance the modality for N of 1 case developments. So with that, I would like to summarize my talk today. We are actively working on our SPTAN1 personalized ASO therapy with a few lead candidate ASOs developed, and we're looking to further advance the efficacy and test for functional rescues in patient-derived cell lines. Sorry. We are learning from the SPTAN1 experience to expand to other N of 1 therapy developments, and there are many other N of 1 ASO-based therapy development projects that are ongoing within our lab. And as a diagnostic lab, we are investigating new modalities for our involvement in the personalized therapeutic field, as I've proposed previously, and hopefully this could be inspiring to other researchers or clinicians in the field so that we can all work together to further advance the N of 1 therapy development community. And with that, I would like to thank members of the Liu Lab, my thesis advisory committee, the Genetics and Genomics graduate program at Baylor College of Medicine, our collaborators, our funding sources, as well as our participating patient families for making all of these work possible. And I'll take any questions. Thank you.
Wonderful. Thank you so much, Christiana. Please feel free to start dropping the questions in the chat. And I'd first like to start with saying, wonderful presentation. I think it was fantastic that you showed the whole pipeline and the whole pathway because we don't talk about it enough. Like really going from the functional validation, what's the pathomechanism, identifying this mechanism of action, and then going like, what's the therapy, testing the therapy. And I really love and appreciate that you showed all the pitfalls and all the things, considerations that you had and where you had to go. So, as I'm waiting for the first question, one of the things you mentioned is the ASO design and the difficulty of doing it manually. And I think right now we unfortunately do not have any tools available yet. I know people are working on them, but maybe you can say something about that you decided to go for the variant in the gap instead of the wings, because I know there's like two groups in the field. So maybe you can tell me what was your thinking about that.
Yeah, so yeah, obviously, so there's I guess two ways of doing this is you can either position your variant all the way between either within the wings all the way through, so including the wings and the gap, or positioning the variant only in the gap. My rationale for that was within the gap region, you actually have so that the active RNase H is recognizing and cleaving within the gap region. So positioning a variant in the wing will only allow for binding specificity. That's the only thing that you can really do with the wing region. But if you position it within the gap region, you not only do you get binding specificity, you also get cleavage specificity. So that's the rationale behind that. Um, there are there are papers out there that kind of did many ASO screening and position it either in the wing and in the gap, and looks like the trend is within the gap. It seems to work a little bit better, but obviously, without a designated study to look at, wing versus gap efficacy, it can vary depending on the situation. Yeah.
Great. Thanks so much for kind of going into detail and explaining this. I have not gotten any questions yet. Are there any questions directly now that people would like to raise their hand and ask something specific? I can keep on asking questions if everybody else doesn't want to. I think it's great that you also discuss like where are we going with this and how long is this gonna take? And now you're of course talking specifically about SPTN1. So meaning if I have another patient, you'll be faster. What's your take on other diseases? Because I think it will really depend on what's the disease and how is it like expressed. Do you have any ideas how long this might take to set it up?
Yeah, so again, as I mentioned previously in a talk, is depending on what information is available in terms of the disease mechanism and the functional assays that are available. So if you have a disorder that, it's been well characterized, what would be a good functional rescue indication. Like if you have data on electrophysiology, calcium indicator assays, that type of work, it's fairly straightforward for a research group or a company to move forward with identifying a lead candidate. And testing that in a functional with a functional rescue. But if you're coming up with a variant that has zero, like you only know it's a pathogenic variant, and the disease mechanism is sort of a predicted disease mechanism, and there's no like cellular evidence on what the expected cellular phenotype would be, what functional consequences could be. It could take much longer, like we have in the case for SPTAN1. We did spend a lot of time on disease mechanism and trying to pinpoint what would be the cause of the seizure phenotype, how is the mutation interacting with its binding partners in a neuronal lineage. So Our estimated time frame can be, roughly around a year for those that have a very clear disease phenotype and a good functional readout to 3 years roughly for a variant that has, minimum knowledge in the field with unknown disease mechanism and no known functional readouts.
Great. Thank you so much for elaborating on this again. There's another question now from the audience, and the question is if there were any differences in the passive uptake, so the genetic uptake of the ASOs between the patients and the controls. Did you see anything?
Yeah, so that is another way of testing for ASO specificity is if we have an allele-specific ASO, we could test it in a patient cell. So the patient cells would have the targeted allele and the control cells won't, depending on which SNP that you're actually targeting. So this is not a mutation-specific ASO, so then it's really dependent on the genome. So if you choose a control which does not have the SNP of interest of target, you should see the expression changes for passive uptake of the ASO targeting the mutation to be basically the wild type allele of your patient cell line. We haven't done that yet because our patient cells are heterozygous, so you're able to see the uptake or the knockdown effect of your ASO to the wild type allele versus the mutant allele. But if you're in a situation where you're looking at a homozygous knockdown, you will probably want to test that in a homozygous non-targeted cell line, if that's what the question is asking. But in terms of passive uptake efficiency, that shouldn't vary between the cell lines unless, there's an uptake defect that we're expecting. And this is a structural protein, so we don't expect that to happen.
I think that's a great answer. And coming to your last point, I kind of want to add that we have seen this actually. So there's diseases that have problem with endocytosis, so really like taking up. And there you can't take up the ASOs, so they can't probably work. So it will work when you do the lipofectamine transfections, but if you trace the genetic uptake, it won't work anymore. So it's a very good point also to think about when you're working with a disease like this. Does it even work in in a natural environment? Okay, any other questions? And I think Christiana, as we said, she agreed to also answer more questions on connections. As if there's any specific questions on cases, please feel free to drop them at connections and then we can look into them and give you some answers. And if there's no more questions for now, I'd like to thank Christiana again for this very interesting and informative presentation, everybody else for attending, and I wish everybody a nice day or evening. Oh, now the light goes out. Fantastic timing. Have a great day and a great week, everyone. Bye bye.
Thank you.
Thank you.